Okay, after that, if this make F1 new generations, then again breeding continues like this until they see all the desired characteristics. They have to maintain as a hybrid like this as a new breed.
So, hybrids increase genetic fitness due to increase of the variations, no, sir?
Yeah, because it is coming from two strong genetic background. So people do this not just for mixing all the pure lines. So they have to first identify the good characteristics, and when they mix these two characteristics, more genetic variation brings to the hybrid. So then, for example, disease resistance. So it is something like this. It's a more than what we thinking. It's not doing randomly. So the people, or now it come to the scientists or people working in the science or agriculture, they are more intelligent. They know the theory than the farmers, so they can study well. After studying well, then only they start to develop this hybrid. So when they study, for example, if they find one variety of plants, they are disease resistance and they get a more yield, and their food quality is tastier. And the second hybrid they found, they are resistant for the drought, and they are resistant for the pesticide, and that is giving seedless fruits. When they identify the large characters like this, then they mix this. So that means you can understand these are all phenotype or alleles. So it's a mixing of large alleles and phenotypes to make a mixture. So this mixture has so much of advantage now. It's like a likewise. Then they maintain one line like this. After maybe many generations, many years, they have a hybrid version. So then hybrid version can again use this one to mix with another hybrid. So this is never ending story. We can go forward with all different inbreeding, outbreeding, and the hybrid, like all things. When we see end organism, plant or animal, they have a desired characteristics. We can again mix two different genetically backgrounds or similar background or hybrid like this. Uh, what is common for both one species? So inbreeding, outbreeding, and hybrid, still we use one species. So we haven't mixed up the species yet. So we use only one species. That's one important thing. So all these three breeding techniques, we are using one breed, one species. Within the species, we enhancing the quality. So it's kind of a experimental method. So let's say now we have a low fat milk. So if we want to make a low fat milk to the market, we can understand they have to remove the fat from the milk. Removing fat is additional cost, and this milk is cost than the full fat milk. Then people are thinking, okay, they're going to find a cow which fat percentage is less. Naturally, fat percentage is less. Then they try to use these cows to make a inbreeding or outbreeding or hybrid whatever method to producing a line which giving a low fat milk. Then they can directly send to market without additional step of separation the fat. So after identification of good characters and the desired outcome, then the breeding experiment practice until it maintain a pure line. So after many generations, scientists can make a pure line of crops or animals.
So that means they hybrid them and they get a what they get the what they want from that, and those they maintain as a pure line. No? So like disease
Yeah,
resistant.
that's the way, because if they like, we can understand like our breeding technique, like probabilities and everything. So if they start the first experiment, let's say they want to make this these two breed or varieties, they're going to have a breeding, and uh, our understanding is at the first, the birth or the first offspring, maybe not the desired one. So after many times, maybe they select this one. So likewise, they have a selection after select this one and these two, and they're going to breed. So likewise, they keep on breeding until they see a desired character. So when they see desired characters, at one point, then this goes as a pure line. So in animal, it might take a long time, because their birth rate and the period of development period is longer than plants. It takes long time to develop a pure breed.
But it's
And also, this is practice in research and diagnostic purpose. If scientists use lab animals, rat or mice, again, companies selling these rat or mice use this kind of breeding techniques to maintain the pure lines. So pure, like even rat, we can find different uh, breeds. They maintain as a pure line, and they have a different qualities, because one rat is easy to handle. They are friendly. So when they identify this character, it's easy for scientists to use these rats, because it's very easy to handle them. Some rats are aggressive, difficult to use. So when they identify this character, they try to maintain pure line with this kind of desired characters. So inbreeding, outbreeding, and hybrid uh, come as a three different method. At the end, desired characters amplify until all the population get this kind of a characteristics. So end, increase the yield, increase the quality, increase the end user satisfaction, aesthetic value, and other characteristics of crops or animals we can expect. Right, the last breeding technique, interspecific breeding. Now, for all these three happen within one species. Now we are going to mix up two different species. Fourth one, interspecific breeding. This may be some kind of iconic picture that we can understand a lion and tiger if they breed, how it looks like. Okay, we take one example of plant. Now within the berry family, like a strawberry, what we know very easily, there are different species within that berry family. So the boysenberry berry is result of mixing of a few different species. So they mix blackberries, raspberries, and loganberries. So this is possible, especially in plants, not like animals. Uh, when we talk about the plants fertilization, they don't have a heavy restriction like animals. So different gametes of different species we can mix. If the mix is quite strong, then its phenotype is mix of all these. So usually we write this uh, interspecific breeding new species. Like first, this rubus is uh, the first one is a genus, and this one is the species name. So it is possible easily within the same genus, right? So here is a rubus, and here is a rubus, and again rubus. So we know that when in classifications, the species are the most biological, smallest one and most active one, they can interbreed. But when it comes to genus, there is a less chance of interbreeding, and they don't appear as a species then. So we separate to genus to species because they can't interbreed. But when it comes to the plant, this restriction is not much strong. We can use this breeding technique within different species in the genus. So scientists are trying now to mix up these gametes to produce new varieties or new species. And the new species we write like this. The first Rubus ursinus is the first name, and there's a cross in middle. And the second one, Rubus idios. So this is the scientific name given for this interspecific species. So it has two scientific names coming from their background of the original backgrounds. And then this one is also coming from interspecific breeding. The Loganberry is hybrid of the blackberry, North American blackberry, and the European raspberry. So it's a quite strong mix of many different species to bring this boysenberry. So again, scientists are trying to make different varieties, new species. So at the end, outcome is yield is high, quality is high, end user satisfaction is high, aesthetic value is high. So scientists wants to keep these features, and they study um, the the chromosomes numbers and their outcomes and different phenotypes. After that, they are practicing these breeding techniques until they get the desired phenotype. So now it become not the farmer's hand, now it become a scientist's hand. So due to this food crisis, scarcity of food, the scientists are working many places in the world to make new varieties, disease resistant maybe, and the yield is high, good tolerance of the environment, because climate change is inevitable now. It is very difficult to stop climate change. More and more projects are coming up to increase the climate change, the global warming. So when the global warming is the problem, the global temperature increase, as a result, disease spread faster and the temperature is higher in certain places. And adverse weather conditions are also expecting. To fight all these things, scientists trying to develop new varieties of crops that will still provide the food for growing population. And we also have some animals developed in this method. I think a well-known animal is the mule. So mule is the breeding between horse and donkey. Horse and donkey make the mule. And male donkey and female horse make this hiney. And the liger is a male lion and female tiger. And also we have a sonki, zebra, and donkey. So this is the specific breeding. So more plants are more attractive, but animals are not much attractive. There is a problem of animals. So you know what is the problem?
They are not fertile.
Yes, we call it sterile. They're not fertile or they are not sterile. You know why?
No.
If you take a donkey, and the horse, so it results the mule and horse 64 chromosomes, donkey has 62 chromosomes. So this is the full set of chromosomes, and the full set of chromosomes are diploid number. Now these cells undergoing meiosis to form the gametes. So when gametes forms, they get a half a number, 31, and 63, 33. 61 and 32. Now these are the gametes formed by meiosis. So meiosis make the haploid number of chromosomes. Now when they make new phenotypes, the new phenotype chromosome number is 63. So mule has 63. Now the problem is, if mules want to make the gametes for the next generations, this is an odd number. It's difficult to, it's not possible to make the gametes, cannot divide. To make the gamete, to form the gametes, because chromosome number is unequal. They have odd number. As a result, they remain as sterile, and their use is not much promising at the moment. So we just learn as some kind of interspecific breeding in animals, but more than that, use is a little bit of not really helpful for us. But the plant, yes, because plants they don't have a restriction, because they have a special ability to keep chromosome number double or triple or quadruple like this. Animal can't, but the plants, they can maintain different chromosome number. As a result, plants are promising in interspecific breeding. Okay, so we have completed the breeding technique. What is left now, the last topic, genetic principle of this kind of breeding. Now, before that, yeah.
So if two interspecific breeding results in an even number of last one, then is it possible to undergo to be not sterile?
Theoretically, yes. Theoretically, yes. They can form the gametes, but again, we cannot rely on one factor. We rely on chromosome number. So chromosome number is one factor, but there are other factors should also consider whether they can keep on dividing until they make the new species, because uh, species formation, how species form. So you can recall unit 3. Originally, the prokaryotic cells make the eukaryotic cells, and the eukaryotic cells slowly progress into a primitive species. Then all the species today we have coming from ancestral species. That means species formation is natural. Species formation is natural thing. It's not a man-made thing. And the species formation also we can understand the mechanism of natural selection. So natural selection we have learned, if species which have different phenotypes due to the different alleles due to mutations. So DNA sequence change, random process, and they have different alleles, and these alleles make different phenotypes. So they can resist from the environmental pressure. As a result, the Darwin original observation, he found the birds, finches, have some kind of similarity between each other, and he thought it's coming from one common ancestor, but they become different species because of the beak size and beak shape, and like that, become different species as a result of this separation from the environment. So we can easily understand, if one place in this Galapagos Island has flowers and finches, they have a long beak, can live in this place because they can take the nectar. But others with a strong beak and short stout beak, they can't live in this area because it's full of flowers and nectar is the most abundant food. So then you have to move to another place to get the food. After that, they separate physically. When they separate physically, reproductively they are separate. Reproductively separate over a long period of time, they adapt to different environmental conditions, and the breeding happen within these two populations. They become two different species. We can also test if the chromosome number we can get equal number, whether it is possible or not. But chromosome number is a one factor, but other factors should also consider. For example, 20, 23, 20, 23, 23, yes, 23. Yeah, we can get some example with chromosome number become again even number, and then what kind of a species we can get, but they should have again some kind of similarities. For example, human 23. So if we can find an animal which has which can give again an even number, I think we can understand it doesn't work, because it is not the chromosome number. Other factors should also important for the survival of this zygote, the survival of the fetus, and survival of the newborn. So at any place, it should be important to survive, and they should fertile to make the next generation. Right now, we have a small section to finish. Then we should start molecular biology and biotechnology. Unit 7. So have you learned anywhere Unit 7 before?
No, sir.
So you're going to learn it new, right? Unit 7.
Yeah.
Okay, Unit 7 is a little bit bigger section, and chances of asking a question is also a little bit higher, because genetic, if you analyze genetic, uh, we can find one or two MCQs, but in Unit 7, I think it's a little bit more question can be asked. Even essay, structured essay, frequently asking molecular biology and gene technology questions, as the topic is very important nowadays, and it is more it has a lot of application in the future. So that would be one reason. Even in medicine, it is very important topic, diagnostic and disease, understanding the disease. That become one of the important topic. Uh, do you have a complete set of note for this one? Uh, for the Unit 6?
Wrote down sir's notes like.
Yeah, that is actually based on the resource book, and I try to add additional things, mostly highlighted in blue color for the understanding purpose. If the things are not enough to understand, we need more information. Otherwise, I also strongly believe in papers will not ask out of resource book. So I have a feeling like that. There's no chance of asking out of resource book. Only maybe small percentage here and there can be changed, some kind of wording and some kind of small concept. Otherwise, if we completely adhere to these notes and resource book, we can get a good marks, because of the mark is key point. They also use the resource book. It would be easy to get it, but limitation is the number of standard questions, because some of the old syllabus we can't use for certain things. I think same for genetic. Some of the question in the past, they are no longer fitting to the new content. Maybe molecular biology is also a little bit advancing now, updated like 20 years ago. The molecular biology is not the molecular biology we have now. It's a fastly updating field. And it's a little bit bigger chapter. Yes. And any date about the exam? I think O level is fixed now to May, third week of May. So you might have exam in October, November, December time, right?
Yes.
If the papers, if they can finish paper markings at the right time, the last exam. Okay. Genetic principle of breeding techniques. Now we have learned these 4 breeding techniques: inbreeding, outbreeding, hybrid, and the interspecific breeding. Now we're going to learn what is actually happening in the cellular level. So when they use this breeding technique, what is actually happening in this breeding technique? And also, the breeding principles are not only these 4. So we have learned the main breeding principles: inbreeding, outbreeding, hybridization, and interspecific breeding. Now, genetic principle of breeding techniques. We can make a new breeding method if we go on to the DNA level. So if you go into the chromosome level, if you're trying to change from the chromosome level, we can establish new species, new breeds, new varieties, which can again support the concept, quality, quantity, resistance, adaptability, end user satisfaction. Like all what we discuss, we can provide more enhanced quality. For example, briefly mention the third one, CA1, 2, 3. Our third one, genetic modification. So genetic modification, we're going to learn in a bigger way for the next unit, 7. But here we again understand genetic modification is one of the main method we can face this food challenge. So breeding technique we use, inbreeding, outbreeding, it is artificial, but we never touch DNA level. We have no interference with the DNA. It is still, we select natural animals and plants and let them breed like that. But genetic modification is going to the roots, and we're going to change everything. So we have to talk a lot in the future, in the next chapters, genetically modified organism, GMO. How we do this, advantage, disadvantage, example. So as a result, I don't think we have to talk too much in this place about genetic modification, but we just give a small overview. What is this genetic modification? Now, second one, induced mutation. So we have learned mutation is very important to make different alleles as it change the DNA sequence. After that, it can make different phenotype. Now here again, man-made thing, induced mutations. Trying to induce mutation. So what is happening naturally going to accelerate. And first one, that is one of the thing naturally happening in plants, having a more number of chromosomes. Having a more number of chromosomes. So it's called poly. Poly is many, ploidy. So diploid and haploid having a chromosomes we call a ploidy, and poly mean many. So haploid and diploid is haploid is 2, diploid is 2, haploid is 1. Then a poly mean many chromosomes, having many chromosome numbers. So again, interfering the human interfering in plants to establish more chromosome number for better quality, because we can understand if more chromosome, more alleles, more phenotypic characteristics. So if we change the alleles, new allele can give a better functions. Mutations are important. Now genetic modifications, we're going to mix up genes in two different genetic background, like two different species. We're going to mix up the genes. Genetic modification, we mix up genes from two different organisms. So then we introduce more alleles to one organism. For example, if there's a plant, we can introduce a good allele to this plant, and then this plant now breed with this character. So we're going to learn these three breeding principles: polyploidy, induced mutations, and genetic modifications. So hopefully the latter part of this topic is little bit of concept and ideas and too much things, not like not interested as calculating things, making some maths like things. So it's more interesting if we have this kind of thing. Here we have to learn and we have to remember facts and figures uh, about the beading technique. So, okay, I think next time within a half an hour we can finish this topic, and then we can start 7 genetic engineering and molecular biology, which is again very advanced concept we have to learn. Okay, now how human induce polyploidy. So we learned that during cell division, meiosis and mitosis, the chromosomes are separate. So this chromosome goes to this side, this chromosome goes to this side. This is possible because this end there is a centriole and there is a centriole here, and microtubules form the spindle. Microtubules form the spindles. Then microtubules shortening and pull the chromosome to the two end. As a result, it results the cells like this. Now, what is the method of polyploidy? A chemical called colchicine inhibit microtubule formations. So then there is more microtubules. So colchicine inhibit microtubule formation. Now the resultant cell is like this. So it has many chromosomes now. So when it have a higher number of chromosomes, there is a problem that the nucleus is loaded with a large number of chromosomes. So nucleus is loaded with large number of chromosomes. So when nucleus is loaded with large number of chromosomes, the cell size also a little bit bigger. So then we can expect in this situation, the original size is bigger. That it's bigger than the original size. When it has more chromosomes, the cells are bigger and the organs are bigger. So this is called Gigas effect. That means the strawberry is usually bigger. So strawberry is usually bigger than the normal strawberry if we induce polyploidy. So instead of having a diploid, they have a triploid, tetraploid, and even hexa and octa.